Hello students, welcome to the fourth lecture for the chapter Principle of Inheritance and Variation. My name is Mouchandra, so let's begin. So in the previous lecture, we talked about linkage and recombination, the concepts of linkage and recombination. So using those concepts, we'll be doing two practice problems, okay? So given here in the screen is the problem. So the problem states that this is the image of a chromosome where there are 5 genes, okay? And we have to arrange these genes in the ascending order of linkage strength to gene H. So if you recall from the concept of linked genes or linkage is whichever gene having minimum distance. So here in this example, H and I, they are closest to one another, right? The distance is minimum here. So the linkage strength, or we can say they are more tightly linked. The linkage strength is high. So using this principle, we have to arrange the genes, okay, G, E, J, I, based on the ascending order of the linkage strength. Ascending order means low to high. Okay, so let's do it. So it with respect to H, G is farthest, right? So if we were to write lowest linkage strength to highest linkage strength. So with respect to H, G, gene G, it is farthest. So the linkage strength, it will be loosely linked to gene G. Because there's gene G and H, they can easily get separated through the recombination process, right? So then E, gene E, is the next farthest, right? With respect to gene H. Then J. J is the third gene, which is we are going towards the highest linkage strength level. Okay, so it is more closely linked gene G with respect to E. Then I, right? So this is the order which is being asked, or the arrangement of the genes G, E, J, I with respect to the linkage strength. Okay, so this problem was quite easy, right? It is based on the linkage strength or uh, linkage between the two genes, or how probable it is that these two genes will inherit together, or will they get separated by recombination. Then moving on to the next question. So there are 4 genes, A, B, C, D, and they are located on the same chromosome. Okay. So, and we are given the recombination frequencies. Recombination frequency means what is the chance that these genes, gene pairs like A, B, B, C, or C, D, they will get separated from one another due to recombination. Okay. So in a population, say if there are 1,000 people, uh, and they saw their genetic composition, their genotypes, and after doing simple average kind of a calculation, they found this recombination frequency for each pair of gene. So we have to find out what is the most likely order of the genes on the chromosome. So there is one chromosome given to you like this, like in the previous example. Okay. And you have to arrange these 4 genes A, B, C, D when you are only given the recombination frequencies. So you again recall that if the recombination frequency is high, okay, the recombination frequency is high, that means the distance is also high. Distance between between two genes, because of which they are getting separated by recombination quite easily. But if the distance between two genes on the same chromosome is less, the recombination frequency will be less. So using this concept, uh, the recombination frequencies RF, they are given here. Okay, so we have to write the correct order of the gene A, B, C, D. So we should start by looking at the gene pair with the highest recombination frequency. So for the pair CD, the recombination frequency is 48%. So they will be farthest from one another. If this is a line, okay, so I can place C and D at the farthest two locations. Okay, then let's start with C. Which of the genes A, B, okay, which of the genes is closest to gene C? So here we know for BC, the recombination frequency is 15%. For AC, the recombination frequency is 25%. So since we are looking for the closest gene to this gene C, so we will choose this pair and place B at a close position to C, right? Now we know the position of B, C, and D. Now we have to place A. What we see that uh, for this pair AB, the recombination frequency is 10%. That means gene A is quite close, very close to gene B. So we have to place it somewhere close, right, to gene B. So there were 4 genes given to us, A, B, C, D, and this is the order that we have obtained. Then you can see that the remaining gene pair AD, the 23% recombination frequency, which is quite bigger. This region is quite bigger between A and D with respect to AB, right? And also recombination frequency of AC is also given, so it is 25%. So in this way, we have got the order of the gene, which is C, B, A, D. This is the order of the genes located on the same chromosome. So that's it. These are the 2 example problems based on the concept of linkage, okay, and recombination. Now moving on to the next topics that is given in the book. First is, what do you mean by mutation? What do you mean by mutation? So mutation means if you are given a DNA sequence, okay, and within that DNA sequence, some change has happened, and this change is permanent. Okay, so a permanent change or permanent alteration has happened within the DNA sequence, and when this phenomena has happened, it will be called a mutation event, okay, or a mutational event. So we know that the DNA is quite a stable structure, okay, and most of the in majority of the lifetime of a person, the DNA sequence is stable. It doesn't change. It is static. But due to few rare events, this mutation happens. And these mutations, since they are permanent, they are often inherited from one generation to the next generation. So due to change in the DNA sequence, the person often sees change in the genotype, and due to change in the genotype, it can result to change in the phenotype of that person. So in the next chapter, you will learn more about what is a DNA sequence, what is there in that DNA sequence, right? How this change in DNA sequence is coming into the change in phenotype, right? So the biomolecules involved. So these concepts will be covered in more detail in the next chapter, that is molecular basis of inheritance. So in that chapter, we will be reading the processes which are behind this. Okay. Now, you should know, you are aware of the term chromosome, right? And you know that there are 46 chromosomes in humans, and each chromosome is made up of chromatids. And a chromatid is nothing, one string of chromatid is nothing but the DNA along with some proteins called histone proteins, which are present in supercoiled form and they form the chromatid fibers, and these chromatid fibers again uh, condense very tightly, okay, and form chromosomes. And we know that chromosomes can only be seen during cell division. And in normal state, when the cell is not dividing, the chromosomes, it is not in condensed state anymore. It is like a mesh of DNA present within the nucleus. So mutations can be categorized into various types. First is the point mutation. Another is the frame shift mutation. So what happens in point mutation? Okay. So you will study this again in more detail in the next chapter, but in brief, let me explain. So in point mutation, what happens? You are given a DNA sequence. Okay, a DNA is made up of bases like A, T, G, G, C. Okay, and due to a mutational event, say this particular base, it has changed. Okay, changed to what? Say it has changed to T. Okay, so this change in one base pair within the DNA sequence is called point mutation. Let me write: change in one base pair is in DNA sequence is actually called the point mutation. Now, this is what is point mutation. Now, sometimes what can happen within a DNA sequence, say 8D, let's take the same example. Within this DNA sequence, few bases, say these two bases can get deleted. Okay. Or what can happen sometimes, few base pairs can get added, say T, C, get added here, okay, in between. It get added, insertion, it is called insertion. So because of deletion or insertion, the mechanisms which are reading these DNA sequences, they read the DNA sequences and understand something else. The meaning of the sequence completely changed. Okay. So when this thing happened, then it is called frame shift mutation. Okay. So these are the two major types of mutations. You will study more detail, like how frame shift mutation is happening, or how point mutation is coming into, protein level, etc. You will study in more detail in the next chapter. Now, mutations, as I said, these are rare events. It happens very rarely. Like if there are 10 million bases, there's a chance that only one will get mutated. One base will get mutated. So 1 in a million chances there for a mutation to happen. So there are certain causes which triggers these mutations. So these causes are collectively known as mutagens, which are causes of mutations. Okay, so there are various mutagens. Uh, these can be physical factors like UV, right? UV radiation. UV radiation, when we're exposed to UV radiation, the cells, the nucleus within the cells, or the DNA within the cells, they the sequence within, they can undergo some change in a base, or some insertion of some base, or deletion of some base, because of which a mutation has happened. Or it can be due to some chemicals as well, which can cause mutation. Often, few mutations are so lethal that it can cause cancer, various kind of cancers. People have found that various type of mutations result into various kind of blood cancers or liver cancers, etc. So people find in the DNA level the cause of these kind of cancers, because often it is the case that mutations are resulting in a type of a cancer phenotype. Now, in humans, okay, in order to study whether a mutation has inherited, how it has inherited, we cannot do test cross, right? We cannot force two people to meet and all, and it will also take years in order to understand. So it is unethical to do it. It was easy for Mendel to do it for pea plants, but for humans, we cannot do it. So another way of studying the inheritance of characters in humans is through making pedigrees, or what is called family trees, in which we understand how a particular disease or a particular normal phenotype is inherited from one generation to the next generation. Okay, so this is called pedigree analysis. So for pedigree analysis, people use certain types of symbols. And here are the symbols given. A circle represents a female, a square represents a male individual. If we use a diamond shape, it is basically representing an individual whose sex is unknown, often an unborn child or a child who died due to miscarriage. So for those people who we don't know, we couldn't know the gender. So for those people, diamond symbol is used. Then when we are studying a particular trait, one particular trait, remember a pedigree, one pedigree or one family tree can show only one particular trait or one particular disease. So you will find few people are affected or showing that particular disease symptoms. So they are represented like this, means they are squares or circles, they are filled. Okay, and those who are not affected, they are not the circles or the squares, they are not filled. Then if we are trying to show that two individuals have married and mated, so we show it through a line like this. And if we want to show that two cousins in within the same family, they have married. Okay, so that is called consanguineous mating. So we show it with a double line. Other symbols, this is for showing that two people are siblings, like here they're brother and sisters. If someone in the family has died, we show it with a cross that this person is dead. Then often a pedigree is made by talking to a particular individual. We ask few questions and the answer to those questions helps in drawing a pedigree. So the person who is being interviewed is called proband, or the index case. We show it by using an arrow. And in the during the inheritance of a particular trait, a person can be a carrier of that trait, or that person is a heterozygote for that particular trait. So to show that, we use a small dot. This is a carrier for that particular trait or a disease. So these are the symbols. Here is an example, okay? Example of a pedigree. So how one should read a pedigree. Okay, so here 1, 2, 3, these show the generations. So this particular pedigree shows a family of 3 generations. Okay, and suppose you are this girl, right? In third generation, you are this girl who is not showing the symptoms of a particular disorder, and you are being interviewed, like how are your parents, right? Then whether your grandparents had the disease or not, how many brothers sisters do you have, then how many brothers did your father have, or how many sisters your father had. So like this we have to interview a particular person, and then we draw a pedigree like this. So here this is the affected grandmother who has married this guy and given rise to the second generation. And these two people, they had 1, 2, 3, 4. They had 4 children. Okay, this is how we show the children of these two individuals, male and female. And among the 4 children, 2 were affected by the disease, right? One male and one female. And we saw that this lady, she married another man, and they had their own children. Similarly, this person, this male, has married another female, and they had their own children, 3 children. And now this is you. Say, suppose this is you, and you're being interviewed, and whatever answers you have given, through that, a complete pedigree, okay, with 3 generations is drawn. Okay, so this is how you should read a pedigree, or this is how you should, you know, draw a pedigree. Okay, now in this slide, I'm asking you that from a pedigree, okay, from a pedigree, what all information can be obtained? So first information would be the phenotype or the disorder you are studying, say disorder or a disease you're studying, whether that disease is autosomal or sex-linked. What do you mean by autosomal or sex-linked? It means whether the mutated gene is present in the autosomal autosomes, in humans there are 22 autosomes, or whether it is present in one of the X or the Y chromosome, Y chromosomes. That is whether it is present on the sex chromosomes. Okay, and how the mutated allele say, how does it behave? Is it a dominant allele or whether it is a recessive allele? So if it is a dominant allele, the person heterozygous for that dominant allele will show the phenotype, whereas the person who is not carrying, okay, not carrying the dominant allele, that person will be normal. But if that allele, mutant allele, is recessive in nature, then uh, for the phenotype or for the disorder to happen, the person should have both copies of the mutated allele. So this is what you have understood from the Mendelian disorder, Mendelian inheritance. So those principles are used while analyzing a pedigree. Okay, so pedigree analysis is done for Mendelian disorders where uh, Mendel's laws of inheritance can be applied, whereas this analysis, pedigree analysis, is not at all useful for those kind of disorders where some change in chromosome structure or etc. like abnormality in chromosome is there. For those kind of disorders, pedigree analysis is not done. Okay, and as I previously told, a pedigree analysis is done for identifying the inheritance of one particular gene. Okay, so if you are studying, if you want to show two disorders in the same pedigree, that you cannot do. You have to draw two different pedigrees for showing the inheritance of two different disorders. Now, coming to Mendelian disorders, as I told, Mendelian disorders are those disorders which follows the Mendel's laws of inheritance. And these disorders are happened or have been reported because of mutation or alteration in one single gene. Okay, so these are monogenic disorders, you can say. There are also those disorders you have already know that they are polygenic, right? More than one gene participate in manifesting those this kind of disorders. So for monogenic disorders or Mendel, they come under, they are like studied under Mendelian disorders, and these Mendelian disorders can be studied through pedigree analysis. So there are various examples of Mendelian disorders like hemophilia, cystic fibrosis, then sickle cell anemia, okay, color blindness, phenylketonuria, thalassemia, etc. So these are all the examples of Mendelian disorders where people have found a mutation in one gene in either autosome or in either sex chromosome, which has led to disease symptoms. Now, first, let's talk about each of these examples one by one. First is the disorder where person is not able to distinguish between colors. That is the color blindness. So this color blindness disorder is actually a X-linked disorder, X-linked recessive disorder, which means the a gene which is mutated is present in the X chromosome, and that mutant allele is recessive in nature, which means affected person will have two copies of the mutant allele. Then only that person will show the symptoms of color blindness. So that is what it is written. The person with color blindness can't discriminate between red and green color. Okay, it is very difficult for them to distinguish between red and green color. It is because there is a defect in a gene which is present in the X chromosome. So in the eyes, there are rods and cone cells. Okay, so in the color blind person, the cone cells for the red and the green color, they are not functional properly, because of which that person cannot discriminate between these two colors. And the gene responsible for giving the cone cells that color discriminating capability, that gene is mutated. Okay, it is that gene is not at all functional. Okay, and this gene is present on the X chromosome, that is why it is called X-linked, and the mutant allele is recessive in nature. So this is an example of a pedigree. This particular image is the example of a pedigree for an X-linked recessive disorder. So if we ask the colorblind person, okay, and ask him questions like how many brothers do you have, how many brothers your fathers have, or whether your grandfather had the disease or not. So asking these type of questions, we can draw a pedigree, and we will get a pedigree like this. So from this pedigree, we can make some conclusions like the X-linked recessive disorder, like color blindness, mainly affects the males. So in this pedigree, you can see this guy and this guy, these two males, they are affected. And females, they are usually carriers. So here, see, these females are carriers. And it is quite rare for a female to be affected. Why? Why only the males are mainly affected? So as I told you, that this is a recessive disorder, and the recessive allele is present only on the X chromosome. So in males, have one copy of X chromosome, one copy of Y chromosome, right? And females, they have both copies of X chromosome. So this is a homologous chromosomes, right? XX. So if the recessive allele is present in X chromosome, there is no homologous pair to, or there is no dominant allele in males, which will, shield the effect of the mutant allele. So in these individuals, that is males, recessive allele, only one copy of recessive allele is sufficient for the expression of this particular disease. Okay, whereas in females, females are often carriers. So if the female has received, say, in this particular X chromosome, the female has received the mutant allele, the normal gene is present on the other X chromosome. So because of which the recessive allele will not get expressed. And affected males, they are often born to unaffected parents. Okay, so this is the affected male, and he has two unaffected parents. Okay, since it is recessive, and daughter can only be colorblind if father is affected and mother is a carrier. Then the X chromosome carrying the mutant allele from the father will be inherited to the daughter, and the X chromosome carrying the mutant allele from the mother will be inherited to the daughter. Now the daughter will have two copies of the recessive allele. So because of it, she can develop color blindness, but that is quite rare. Okay. Now, another Mendelian disorder is hemophilia. Now, hemophilia is also a X-linked recessive disorder. So the pattern of inheritance is similar to this particular pedigree. Like we can draw a similar pedigree for hemophilia as well. Here also the same features apply, like the males are usually affected, the females are usually carriers, and affected males, they are born to unaffected parents often, and where the mother is a carrier. And what happens in hemophilia is a single protein which is part of the blood clotting cascade. So in the blood clotting cascade, there are 8 proteins, and one of the 8 proteins is mutated, because of which that protein is not functional. So the blood clotting cascade cannot function properly, because of which the person will show non-stop bleeding, even if that person gets a very small cut, because and the person does not have the clotting ability in his blood to heal that small cut. So these people with hemophilia, they have to be really um, protective. We protect themselves from sharp things so that they don't get cut easily. So as I told you, this is X-linked recessive disorder. So it mainly affects the male. So only one copy of recessive allele is sufficient for manifestation of this particular disease. Then females will be affected only if mother is a carrier and father is hemophilic. Okay, so this is quite rare, by the way. This is quite rare. Now, another Mendelian disorder is sickle cell anemia. All right. So this is now a autosomal recessive disorder. Means the mutant allele is present in the autosomes. Okay, and one of the autosomes, and the mutant allele is recessive in nature. So this is an example of pedigree, which can be drawn for autosomal recessive type of disorder. So here you can see that the affected person can be either male or female. Okay, and because the mutant allele or mutant gene is present in the autosomes, and both male and female, they have the autosomes, and the parents of the affected people, they are actually carriers. Okay, and they are not showing the symptoms of that particular disease, and it can affect either sex, as I told you. So for this sickle cell anemia disorder to happen, the gene responsible for this disorder, it has 2 alleles. Okay, these two are the alleles, HBA and HBS, where HBS is the mutant allele, okay, which is having the mutation, and HBA, this is the wild type allele or the normal type of allele. Okay, so the affected people, they will have a homozygosity for HBS kind of allele. Okay, because this is a recessive disorder. For recessive alleles, has to be in homozygous condition for the expression of this particular disorder. So the affected people, they will have a homozygosity for HBS allele, whereas the person who is a carrier, they will carry one copy of HBS and one copy of the wild type allele or the normal allele, which is HBA. So in sickle cell anemia, it happens because of a point mutation event where a DNA base A is changed to T. Okay, this is a point mutation event. And where it happens in the 6th codon of the beta globin gene. It happens in the 6th codon of beta globin gene. So beta globin is one of the polypeptide chains which make the hemoglobin molecule. In hemoglobin molecule, there are 4 globin chains, 2 beta chains, and 2 alpha chains. So one of the beta chains carry this point mutation. So because of this point mutation, the resulting amino acid also gets changed. How? The glutamic acid. Okay, glutamic acid is one an amino acid which is normally present in wild type. It is substituted to the amino acid valley, is present in mutant people, like mutant type. So the HBS allele will be having this point mutation, and when that allele is expressed into a polypeptide chain at the 6th position, valine will be present instead of glutamic acid. Now this mutant hemoglobin molecule, okay, which is carrying this mutated beta globin protein, uh, this it will result in change of the shape of the RBCs, red blood cells. So red blood cells, which are normally they are circular and biconcave, these RBCs change their shape to mutated to sickle type, like a sickle shape. This is the sickle shape. Okay, so the normal biconcave circular shape of RBC is changed to sickle shape of RBC. So because of this change in shape, um, the RBCs are not able to carry enough oxygen. Okay, so the oxygen content in the blood is very much reduced. So the person develops an anemia kind of a symptom. Okay, and this transition of the shape of the RBC happens under low oxygen levels. Okay, when the oxygen level in the blood is quite low. Now, next Mendelian disorder is phenylketonuria. So, if any of you have a query regarding whatever I have taught till now, please share your queries in the YouTube chat box, and those will be communicated to me. I'll have a look and answer those queries for you guys. Okay, feel free to write your queries. Thank you. So the next Mendelian disorder is called phenylketonuria, and this is a metabolic disorder. Means a metabolic pathway is not functional in people having this disorder. So this disorder is autosomal recessive, just like sickle cell anemia. Means a defected gene is present in the autosome, and the mutant allele is recessive in nature. The affected individual who are homozygous for the mutant allele, they lack the enzyme phenylalanine hydroxylase. Okay, so this enzyme is responsible in normal condition. It converts phenylalanine amino acid to tyrosine. Now, since the affected people, they do not have this enzyme, this conversion doesn't happen, because of which phenylalanine accumulates. Okay, it accumulates within the cell, and while it accumulates, it gets converted to phenylpyruvic acid. Now, this phenylpyruvic acid is very toxic to the cell. So, if this accumulation is happening in the brain cell, the affected individual shows the symptoms of mental retardation. Whereas if this accumulation of phenylpyruvic acid happens in the skin, there is reduced pigmentation of hair as well as skin. So this particular disorder um, is due to defect in one gene, but it is showing multiple uh, phenotypic expressions, right? It can affect the brain, it can affect the skin. So this disorder can come under the category of pleiotropy, which you are already aware, I think. So it was taught previously. So let me recall, pleiotropy is a phenomena when a single gene exhibits multiple phenotypic expression. So here the mutation in one gene is resulting in defected phenotype expression in both brain and hair, and both these type of phenotypic expression are completely unrelated. Okay, so they are happening on their own in two different tissue types. Now, this is the last kind of Mendelian disorder that I'll discuss today. It is called thalassemia. Now, thalassemia is again an autosomal recessive disorder. Okay, now here what happens, the gene responsible for thalassemia undergoes mutation such that there is a reduced rate of synthesis of one of the globin chains of hemoglobin. So as I told, hemoglobin have 4 chains, 2 alpha chains and 2 beta chains. Now the mutation can be in the gene which encodes for the alpha globin chain, or it can be in the beta globin chain. So because of this mutation, there is less synthesis, less amount of alpha globin chains synthesized, or there can be less amount of beta globin chains present. Okay, so which hampers the normal functioning of the hemoglobin molecule. Now, such people having thalassemia, they have abnormal hemoglobin, because of which they cannot carry the oxygen in a normal fashion. Less oxygen they can carry. So because of which anemia kind of a symptom is seen in people having thalassemia. Now, thalassemia can be of two types. It can be either alpha thalassemia or it can be either beta thalassemia. So in alpha thalassemia, what happens? Two genes, mutation in either gene ABA1 or ABA2. Mutation in either of these two genes takes place. Okay, and these two genes are for encodes for alpha globin gene. Okay, and these two genes are found to be present in chromosome 16. Whereas beta thalassemia, this type of this variant of thalassemia, this happens because of mutation in a gene that is called HBB gene, and this gene is present in chromosome 11. So HBB gene in normal person codes for the beta globin gene. Okay, so the mutation results in defected production of beta globin gene. That is defect. What is the defect? Reduced. Reduced amount of beta globin chain will be produced, because of which abnormal hemoglobin molecules will be formed. So this is all about thalassemia. Once again, if you have any queries about all the Mendelian disorders that I have discussed so far, please write those queries in the YouTube chat box. Now coming to the next type of disorders, which are chromosomal disorders. Okay, so here what happens, the disorder is seen because of uh, abnormality in the chromosome structure. Okay, so what kind of abnormality? First is aneuploidy. In aneuploidy, what happens? A person, the affected person, either gains an extra copy of the chromosome or it loses one copy of the chromosome. It happens because during meiosis, if you recall, during meiosis, these are the, this is one chromatid, this is another chromatid, and this is one chromatid, this is another chromatid, and these are called sister chromatids. So during meiosis, during normal mitosis as well, that happens at the center, the chromatids have to separate. So when that segregation doesn't happen, when there is a failure in segregation, what happens? There is a gain of additional chromosome, okay, in the gamete. So if the gamete which are formed, I'm drawing here, the gamete which are formed, instead of having one sister chromatid each, one gamete has both sister chromatids, whereas another gamete doesn't have any sister chromatids. It is empty for that particular chromosome. So now, if these defective gametes go ahead and fertilizes another male or female gamete, it nature allows the development of that zygote for development, and that it becomes a person. So those person will show a disorder. That those person will not be normal like you and me, right? They will show some kind of disorders. So people have categorized these disorders, like they have found that people often have this kind of disorders like Down syndrome, Turner syndrome, Klinefelter syndrome, etc. So these all three syndromes happen because of either gain or loss of chromosome that happens due to failure in segregation of these sister chromatids during meiosis process. Now, another event that can result in gain or loss of chromosome is polyploidy. Okay, where complete set of chromosome is added. Okay, and it usually happens in plants. So if suppose the plant, normal case, the plant is a diploid. Okay, now one complete set of chromosome, that is one haploid set of chromosome, is added. So a mutated plant or the affected plant will have a genotype of say 3N. Okay, it can have 4N, 5N, like this. So this type of condition results into polyploidy, and they will show some symptoms. Okay, so why polyploidy happens? It happens because towards the end of mitosis, telophase happen, and then cytokinosis. So cytokinosis results in division, right? Formation of a cell membrane and dividing the two daughter cells. Now, if that cytokinasis doesn't happen, so it will then form one single cell where the two sets of chromosomes will be present. So in those kind of situation, polyploidy results, okay, and symptoms, disorder symptoms results. So let's look into each of the aneuploidy related syndromes, the syndromes which are happening because of aneuploidy. First is Down syndrome. Now, what happens in Down syndrome? The person gains one additional pair of 21 chromosome, because of which that person has three copies or trisomy, or three copies of 21 chromosome. In normal person, there are two copies of 21 chromosome, but in Down syndrome, person with Down syndrome, they have three copies of 21 chromosome. And these people, they often have very short height, okay, short statured, with small head, small round head, and in the tongue you can found furrows or holes kind of things. Okay, the tongue is not plain smooth like us. Also, these children or these people, they often have their mouth partially open. They cannot close their mouth or keep their mouth closed at all times, and they often shows default in physical and mental development. Okay, so this is about Down syndrome, where the affected person has three copies of 21 chromosome, or it is also called trisomy of 21 chromosome. Next, another syndrome which happens due to aneuploidy is Klinefelter syndrome, where the affected person has one extra copy of X chromosome, because of which instead of 46 chromosome, the person has 47 chromosome. Now this individual is female, okay, but because of the presence of Y chromosome, this individual develops masculine characteristics like a man, okay, but this individual is sterile, means they cannot have children of their own. Her female reproductive organs, they are not properly functional. Now, another syndrome is Turner's syndrome. All right. So in Turner syndrome, what happens? The affected person lacks, okay, lacks one copy of X chromosome, because of which I have written this 0. So one copy of X chromosome is absent. That is why that person has 45 chromosomes instead of 46. And this person is again a female, and she is sterile. She cannot have children. And because of the absence of one X chromosome, this person shows underdeveloped feminine characters. So that's all for today's class. Today I have finished the chapter Principle of Inheritance and Variation. I hope you guys can go ahead and read the chapter in the book. And if you have any queries, you can always post them in the YouTube box. Thank you.